Bismillah Rahman Rahim. We now discuss the paper 6 because that's in a few days, and some of you have commented and you would like some sort of a guidelines on how to solve the paper 6. So here are a few tips for solving the paper 6. As you all know, this paper is for 1 hour, and this is 5090 paper 6-2. So you must remember the time frame because some of you think it's for 1 hour 15 minutes. Well, that's not the case. It's only for 1 hour, and you must try and finish both the questions, or sometimes there are even three questions in this 1 hour. Start with question number one. In the beetroot plant, cells in the root contain a red pigment. First, always read the question very carefully and read it twice. This pigment remains in the cells of the plant cells unless the cells are damaged. If beetroot tissue is placed in water and the cells are damaged, the pigment leaves the cells and colors the water. So this is the plant cell, and in this we have a pigment. But if this is damaged, the pigment is going to leak out. So understand the question first. Some students investigated the effect of temperature on the release of the red pigment, which means the temperature effect of temperature on how much the cells will be damaged on the release of the red pigment from the beetroot tissue. They followed these instructions. Read this question twice at least. Cut 4 cylinders beetroot tissues, each 30 millimeter into 10 millimeter. 1, 2, 3, 4. 4 cylinders. Just like, when we make chips or fries, you cut them into cylinders. Wash the beetroot cylinders in the beaker of rinsing water. So you have a beaker of water, and all you have to do is you have to rinse them. You have to just wash them in this. Not just rinse them means not wash them with any soap or anything, but just rinse them in this. Then you labeled 4 test tubes A, B, C, and D. You labeled 4 test tubes A, B, and C. You haven't put anything in them. Now place one beetroot cylinder in each test tube, and I've just done that for you. So I placed this one here, one here, one here, one here. Nothing's in the test tube as yet. Nothing. It's only the beetroot cylinder. Place test tube A. Place test tube A in a beaker of water at 20 degrees Celsius. So you placed it in another beaker which had water at 20 degrees Celsius. Then you made 3 water baths at initial temperatures of 40, 60, and 80. So you again added another 3 water baths, 40, 60, and 80. Water baths means another additional beaker with water at this temperature. B had 40 degrees, C had 60 degrees, and D had 80 degrees. Now I've written all those here and I've made the diagram for you on the side. And you can do this on the exam, you can do it on the paper. Add water from each of the water baths. Now what you had to do is you had to add water from each of the water baths. So you took this water, I'm using a purple color. So you took this water outside, which was at 20, and you put it in this beaker, in this test tube, sorry. So you add water from each of the water baths to the test tube placed in it until the beetroot cylinder is just covered with the water. So you put this 20 degree Celsius water and you just cover it up. The water in the beaker was at 20 degrees. Add water from each of the water baths to the test tube placed in it until the beetroot cylinder is just covered with the water. Leave the beetroot cylinder for 5 minutes. So you added water A, then you B, then C. So B was at 40, C was at 60, and D was at 80. And then you left the beetroot cylinders for 5 minutes and then remove the cylinders from the test tubes. Then you removed them. So you removed them from it. You removed the cylinders. Now, what you need to do is you need to look at the color of the water. Now you need to look, you've removed the cylinders. Now you need to look at the color of the water in it. Now the color of the water in it could be a dark red or a light red or a orange or something like that. But then they're telling you the question, place the test tubes in the test tube rack and observe the water in them. So you put them away. Now you take them out of the water bath and you put them in a test tube rack, and this is what you see. The table shows what the students observed. Observed means what did they see? Conclusion is something else. You observed something, but the conclusion means what did you conclude? The color of water was the lightest red at 20 degrees Celsius. At B, it was slightly darker, then 60 degrees Celsius, and then 80 degrees Celsius. Right? So it was light red, so that means the damage was less in 20 and it was the maximum at 80 degrees, because it said when it was damaged, when the cells were damaged, the pigment would be released. So this is how you read the question, you understand the question, you sort of make a little pencil diagram and you explain this to yourself just like I've done this to you. It has taken me 6 minutes to do this to you. Now let's look at the question and read the whole question. I'm not going to read the whole question here, but I'm going to go along. But when you're doing the exam, please read the whole question and then start attempting to do the question. First, read the whole question. Part 1, suggest why the beetroot cylinders were all cut to 13 millimeter into 10 millimeter. That's for 2 marks. Always look at the marks as well. So you've got to give me something like 2 points or maybe even 3 points, because maybe one of them is wrong which you've written. Then let's look at the next part of the question. Another student, sorry, no, here we come back. Part 2, explain why the beetroot cylinders were washed before starting the investigation. Part 3 says, and this was 1 mark, Part 3 says, suggest what the students concluded. Conclusion means what did you sort of conclude? What did your assumption? What did you think was the case? Their observations, and explain your answer, conclusion and explanation. And that's another 2 marks. Then state 2 possible sources of error in the method you used. For each, explain how the method could be improved. So what is the source of error and what is the source of error and how could you improve it? Then B, another student used an instrument called a calorie meter. I'm just going to tell you a little more about that, to obtain a numerical value for the color in each test tube. She did the experiment twice. Explain how repeating the experiments make the observation of color run more reliable. Observations to make the observation of color more reliable. That's another 1 mark. Then some of the readings of the calorie meter are shown. So temperature at 20 degrees, experiment 1, 0.6, 1.6, and then you have to calculate the average, and then 1.6, 2.8 and calculate the average, then 4.9, 4.7 and calculate the average. Now, what did you have to do? Let's look at what is the next part of the question say. The calorimeter reading at 80 degrees for experiment 1 was this. For experiment 1 was this. So, at 80 degrees, the reading was that. Insert this reading into the table of results, right? Calculate the average reading for each temperature and complete the table, and that's for another 2 marks. On the grid, construct a line graph to show the relationship between temperature and average calorimeter reading. Join your points with the ruled straight lines, right? That's 4 marks. Use your graph to determine the average calorimeter. Use your graph to determine the average calorimeter reading for 45 degrees. Show you're working on your graph. Now this was a total 19 marks, and you should have done this. You should have taken, just like I've read it, it's taken me 9 minutes now. So by 9 minutes I've read the whole question, and maybe I need to read it once again. Now you can see that this is a 40 marks paper, so you have to realize that 19 marks, you should have spent at least 20 minutes on this question, and we've spent 9 minutes reading it. So you can see how long it is going to take for you to really solve this paper. Now coming back to part 1, suggest why the beetroot cylinders were all cut to 30 into 10 millimeter. Yeah, naturally we want to compare the results. So you can only you can only change one variable. You were changing the temperatures. So then the size of the beetroot strips should have been the same. If the size had also been different and the temperature had also been different, then of course you couldn't be studying the effect of temperature. Then you would say, okay, one is smaller, one is larger, so that would be different. So then if you wanted to compare the different sizes, then you would keep the temperature constant. But here, because we have changed the temperature, so we are keeping the surface area constant. So to compare the results, so that is the same surface area exposed to the water, and the temperature is the only variable which we have changed. Coming to part 2, it says explain why the beetroot cylinders were washed before starting the experiment. Naturally, when there was this beetroot and you had cut a slice from it, well, you had damaged some of the cells here at the edges, and some of the pigment must have leaked from that. So that is why, to remove the pigment which might be lying on the surface. Why would you wash anything? This is to remove something which is lying on the surface, you would wash it. Number 3, it says suggest why the students what the student concluded from their observation and explain your answer. Well, of the obvious conclusion was as temperature increases, more pigment is released because the darker the color it became. And the explanation was that heat will damage the cells. Maybe it natures some proteins, which of course causes the release of the pigment. So that's what biology teaches you. As temperature increases, more pigment is released. That was the conclusion. Conclusion means what did you sort of conclude? What did you think was happening? So as the temperature increases, more pigment is released. Explanation, biological explanation is heat damages the cells. Then it says state two possible sources of error in the method used. For each, explain how the method could be improved. Now this is, look at it as 4 marks. So you have to be very careful how you answer this part of the question. This is a very difficult part of the question. Now, when you look at these sources of error, state two possible sources of error. Now, so possible they're not all in the water at the same time. You see, you put one piece of beetroot in one test tube, and then you put, after maybe 30 seconds or 40 seconds, you picked up the second one and then put it in the second one, which was at 40. And then after that you put, so it was about a minute before you put it in the third one, and then a minute and a half before you put it in the fourth one, and then you waited for 5 minutes. So somewhere the last one was in it for uh, the first one maybe was in it for 5 minutes, but the second one was for less than five. But then you started the stopwatch at the when you put the first one in. So they were all not in for 5 minutes in the test tube. So if you did them one at a time, like you put in A and then you waited 5 minutes, then you put B and then you waited 5 minutes. So you would have actually done this in 20 minutes. So do one at a time. Then the second source of error was volume of water was not measured. We just added water to cover the beetroot. And that could be a little over it or could be a little just below it, and you just said cover it. I mean, that wasn't basically a very scientific manner to say it. So did we measure the volume of water? We didn't measure it. We just said, okay, add water enough to cover the beetroot. So you should have measured the volume of water. Whenever you're using a liquid, remember the volume is very important. When you're using a solid, then the mass is very important. Like we didn't measure the mass of we just said cut it in this length. But then the water added was not measured, and we just said add water enough to cover it. Well, that could be the volume of water could be different in each of the test tubes. And there was another third source of error as well. So there were three in the mark scheme. There was one, two, and the third one was temperature of the water bath was not maintained. So if the temperature was not maintained, we just kept it at 20, but we didn't use a thermostatically controlled water bath. And this is of course electrically. So this is a word which I like you to use, thermostatically controlled water bath. Whenever you're using a water bath, do write about this. Thermostatically controlled, that means that they're controlled, just like you have an oven in which you make cupcakes. Well, you control the temperature. So temperature of the water bath is not maintained. And then of course you could have said use a thermostatically controlled water bath. Now coming on to the B part of the question, another student used an instrument called a calorie meter to obtain a numerical value for the color in each test tube. She did the experiment twice. Explain how repeating the experiment makes the observations of color more reliable. You see, when you take an average or a mean can be calculated. So average or mean can be calculated. Or you can say mean can be calculated, because if you just take one reading, well, that could be a wrong reading, could be an error. So when you take 2 or 3 or 5 readings, well, then you calculate the mean and you get a very good idea as to how much could there be a change, how much could it be vary. So average or a mean can be calculated, or you can see shows up any anomalies. Now what is an anomaly? Anomaly means a mistake. So those are called anomalous readings. So anomalous readings would help us to decide, okay, well, these three were this, and then there was a fourth one which was very different. So maybe that was an anomalous reading. So if you have five and one of them is sort of not following the pattern, then you know that was an anomalous reading. So please look up, if you are having problems, then look up what are anomalous readings and you can Google search them. Now it says some of the readings from the calorie meter are shown. Now please remember this is calorie meter. Something known as a calorie meter, that's something else. CALORI. That's a measures the calories. Now that is different than a calorie meter. This is COLOR. Now I'm going to just show you what is a calorie meter. The principle of a calorimeter is the technique frequently used in biochemical investigation involves the quantitative estimation of colors. Color can be produced by any substance when it binds with color-forming chromogens. The difference in color intensity results in difference in the absorption of light. The intensity of color is directly proportional to the concentration of the compound being measured. Calorimeter looks like, and you actually put the test tube here which has that colored liquid in it, and you measure the amount of light that is absorbed, or what the light that is transmitted is absorbed and how much is transmitted. So it measures the light absorption. This is the basic principle of a photoelectric calorimeter, and this is a sample holder, or it is called a cuvette. Please understand the spelling, cuvette. And then there's a lamp, and then there's a lens, and there's a diaphragm, and then there is sample holder, and then we measure the amount of light which has passed through, or the light which is absorbed, and how much light has been transferred to the, and that measures the light absorption. So you just put in all the readings that were given to you, and then you had to just calculate the average, and this would of course be the way that you would do this table. Insert this reading into the table of results, calculate the average reading for each temperature, and complete the table. So you had to do it in the previous page, and you have to complete this, and then this would get you your 2 marks for this and another mark for this. So 3 marks, which is just, I think, very easy marks, and everybody should have got this. Then coming on to the graph, on the grid construct a line graph to show the relationship between temperature and average calorimeter readings. The average has been is given in bold letters. So join your points with rules load line. Now, what is the method? What is the main way how you do a graph? There's 4 marks, and there's 4 very easy marks, and nobody should miss these marks. Is the first thing is you label the axis. Now, how do you label the axis? This first column, the first column is the independent variable, and it is always plotted on the x-axis. And that mnemonic is FIX. First column is the independent variable. Independent means that which you change. So you decided to take 20, 40, 60, and 80. So this will go on the x-axis, and this average reading and this label must go on the y-axis. Please always remember you have to write this in full on the y-axis, and you have to give me these average readings, while this, which is the temperature in degrees Celsius, would go on the x-axis. This is always the rule. First column is always the independent variable and goes on the x-axis. Now you can see labeling the axis is very important, and you must write like here instance, for instance, here look, there's a mistake. I haven't written temperature in the units. So if I haven't written the units, I lose a mark. Here I've written the units, arbitrary units. I've written the whole thing. So just labeling the axis gets you 1 mark. Then choosing the scale gets you the second mark, so that you use the entire grid, nearly the entire grid. I couldn't get it 80 up to here. I could have broken the scale and started 20 here, but I didn't feel I could really manage that. And then I plotted it. That's 1 mark. And then joining it point to point. Now please do not extrapolate. Extrapolate, I mean, is do not sort of, extend it to zero if there's no reading to zero. So then you have to connect these. You can do that with a ruler, or you can do a gentle curve. I might not get it very correct on this because my pen is a little thicker. So you do not extrapolate. I mean, do not extend this, and do not extend it to zero. If there's no readings at zero, please do not extend it. So do not extend it. That's called extrapolation. So do not extrapolate. Just plot the points that were given to you here. The points that were given to you, you're supposed to only plot those. So please, everybody, you must not lose any marks in this. You must get 4 out of 4 in the ATP in the graph question. You must label the entire axis. You must choose your scale. 1 mark is for labeling the axis. 1 is for choosing the scale if you get the correct scale, and you must write the scale every 2 centimeters. So you must write the points here, like I've done it on the other side, and I've done it here 20, 40, 60, 80, and similarly on this I've written 2, 4, 6, 8, and 10. And then of course you join the points. And by the way, the method of plotting is you put a dot on a circle. So either you do this, and the dot must be a dot, not a blob. Don't do something like this is a blob, this is not correct, this would be wrong. Or you do a cross, whatever you like, just do that. Either you do a cross or you do a dot with a circle. Now it says use your graph to determine the average calorie meter reading for 55 degrees. So here is 55. I'm sorry, I can't get it very right. 55 degrees. So you extend the line and you see where it cuts this, and then you read off this. This is the reading that you have to read off. So this would be at least 4 arbitrary units. The units is given here. So you have, but the important is show you're working on the graph. Drawing this line was the important thing, the red line, and that is the one which would have got you 1 mark, and then of course the reading it off would have got you the second mark. To question number 2, the photograph shows a section through a human organ. Identify the organ. Naturally, this is the kidney, and name the structure labeled E, that was the ureter. The B part of the question says in the space below, make a large drawing of this organ as it appears in the photograph. So all you had to do was, I always tell you, is that you just trace this. You just make a drawing, make it a little bigger than this. You just trace this out, and this is how you would show it in the diagram. I'm not saying trace it on this, but it should be just like a looks like a tracing of it. And the marks were minimum size, as long as it covers the whole part of the paper that is given to you. Clear and clean outline drawn with a sharp pencil. Detail of boundary between cortex and medulla. So you have to draw this line as well. The boundary between the cortex and the medulla. And ureter clearly drawn with an open end. This is where you have to draw it like this, which showed that open end wasn't closed. And a correct proportion, that the height was greater than the width. You can see the height was greater than the width. I mean, this is a very rough drawing that I've made, and this is all what was expected of you. It should be cover a big of the space provided to you. It should cover nearly 2/3 of the space provided. Clear and clean outline drawn with a sharp pencil. There should be no these broken lines. So if you're writing it, please do not do these broken lines. You cannot allow this. This is wrong. Then detail of boundary between cortex and medulla and ureter clearly drawn with an open end, and correct proportion, height was greater than the width. On the photograph, draw straight line between F and G. So here I've drawn this in red, a line between F and G. So this was F and this was G. So you have to draw a line, then you have to measure this line, and it says record the length, and it says millimeter. Now this is where you got it wrong. If it was 12 centimeter, it was 120 millimeter. But if you didn't convert into millimeter, well, that's where you got lost the mark, because you didn't really even bother to look at the units. So, and then of course, actual length of this organ is 12 centimeter. Use your measurement C1 to calculate the magnification of the organ in the photograph. Space for working. So actual 12 centimeter. So measure and record the length of this line. You have to measure that. However, it was 12 centimeter was 1220 millimeter. This was probably more than that, the picture, because I don't know the actual. So I'll just measure it and let you know how much was the length of it. But this 12 centimeter was, you see, you have to convert it into. So the actual length was what we will record from the photograph. Let's assume that this was 24 centimeter So this would be 240 millimeter. I don't have a ruler with me at the moment, so I'm just doing a rough calculation. So say this was 24 centimeter or maybe 20, and it's so 24 centimeter means it was 240 millimeter. But the actual was 120 because it says the actual length of this organ is 12 centimeter, which means it is 120 millimeters. So if it was 240 millimeters, so 240 divided by 120, and the magnification would be twice. So this would be twice. So this would be 2 times magnified because the photograph is whatever the length divided by the original. So it's the image over actual. Magnification is what? What is the formula? Is equal to image over actual. So image was the photograph, which was 240, and the actual was 120. So the answer is 2. So it was magnified 2 times. And we move on to question number 3. Variegated leaves have green areas that contain chlorophyll and white areas where chlorophyll is absent. A student investigated the factors needed for photosynthesis to take place. He used a plant with leaves that were all variegated as shown below. So there's a green area and there's a white area. He began by placing the plant in a dark room for 24 hours, explained why this was necessary. This is to de-starch a potted plant. No starch is present. So all that the starch is used up when you kept it in the dark for 24 hours. So initially there was no starch present, and then you set up the apparatus and then you see if starch is produced or not, because if photosynthesis is taking place, then glucose is made and glucose is converted to starch. He then picked a leaf, immediately prepared it and tested for the presence of starch, named the testing reagent iodine solution, name a suitable piece of apparatus, either you said a dropper or a pipette. Then it says describe and explain the appearance of the two areas of leaf after he had carried out this test for starch, which he has done initially. So both areas would be brown because there would be no starch present because you've de-starched the potted plant, and the reason is no starch present. Then he says, C part, he moved the plant into bright sunlight for a day and that tested another leaf for the presence of starch. Complete and label the diagram of the leaf to show any colors. So you would, of course, this would be the green would be the black area. Why would it be black? Because there would be starch present and it reacts with iodine to give you a blue-black color. But the rest of it would be brown because there would be no starch present here and the iodine color brown would remain brown. So this is what you would get, the 3 marks. It says leaf drawn with two distinct areas, central area indicated as black and the outer area indicated as brown. Now state your conclusions from the investigation. So what were your conclusions from this investigation is that light is necessary and chlorophyll is necessary. So your conclusion from the investigation about the factors needed for photosynthesis was light and chlorophyll is necessary for photosynthesis. The green part contained chlorophyll. When you exposed it to light, the green part chlorophyll contained green, so it was necessary for photosynthesis, and that got you your 1 mark. This completes your paper on ATP, and I hope this has been a helpful exercise. And I hope if you have anything more which is not clear to you, please do leave your comments, and I hope to help you as much as I can. Thank you very much for watching.